Okay, so I was just looking through the introductory text for our session today, specifically focusing on the molecular basis of inheritance. And honestly, this completely mind-blowing historical juxtaposition just hit me.
Oh, yeah. What jumped out at you?
Yeah.
And let me just quickly formally welcome you, the listener, to this deep dive. We've got a really foundational topic today. But yeah, what was the realization?
Well, we spent all this time studying Gregor Mendel, right? He's the foundational figure. He mapped out all those statistical rules of inheritance.
Right, the mathematical precision of the P plants.
Exactly. But reading this chapter intro, the reality of his situation is just stark. Mendel had absolutely no idea what DNA was, like zero concept.
Not at all.
He was observing phenotypic ratios and deducing the existence of underlying what he called factors, but he was operating in a total physical vacuum. It's like he was describing a software program without ever having seen the computer it ran on.
That is a fantastic analogy. And honestly, that historical gap is the perfect entry point for what we are doing today. Because what we are looking at is the transition from abstract genetics to actual physical reality.
Right.
For decades, science had the rules of inheritance, but they completely lacked the physical substrate. So the mission of this text, and our mission for this deep dive with you today, is to map exactly how biology bridges that gap.
Yeah.
We are answering the most foundational question. What is the literal, physical, molecular basis of life? When a trait goes from parent to offspring, what physical object is actually moving through space and time to carry that information?
Yeah, and the text highlights just how long that gap in knowledge lasted, which is wild. It notes that at the time of Mendel, the nature of his proposed factors was entirely unclear. And then it says, let me quote it, "Over the next 100 years, the nature of the putative genetic material was investigated."
100 years.
A full century, a century between knowing there is a genetic code and actually finding the physical molecule that holds it.
And you know, the phrasing there in the text is really critical, especially for anyone studying the history of science. Specifically, the use of that word putative.
Putative, yeah.
Putative just means generally considered or reputed to be. Because for 100 years, scientists were basically hunting for a ghost.
They knew it had to be there somewhere.
Exactly. They knew a physical material had to exist because information cannot be transmitted without a physical medium. You can't pass a message without a sound wave or a piece of paper or a stream of electrons.
Right. You need a thing.
You need a thing. In biology, you cannot pass a trait without a molecule. But identifying which of the thousands of molecules in a cell was the actual putative genetic material that required generations of meticulous elimination.
Which makes total sense when you just think about the sheer complexity of a cell. If you grind up a cell, you have lipids, you have carbohydrates, proteins, nucleic acids. It's all just mixed together in this soup.
And for a long time, proteins seemed like the most logical candidate because they are so complex and varied.
But the century-long investigation culminated in a completely different realization. The text establishes that the genetic material is actually DNA. Deoxyribonucleic acid.
Right. And that realization forces us to rely on our foundational chemistry. The text actually assumes a prior understanding from your previous coursework listener. It specifically notes that nucleic acids are polymers of nucleotides.
Yeah.
It is absolutely crucial to internalize the structural necessity of a polymer for information storage.
Okay, let's unpack that for a second. Because if we look at the chemistry of it, a simple molecule like glucose or a straight lipid chain, they can't store complex sequential data.
No, they're too repetitive.
Right, they're too uniform. The requirement for a genetic material is that it must be capable of near infinite variation to account for all the diversity of life on Earth.
Exactly.
But at the same time, it has to maintain a stable, predictable, overarching structure.
And that right there is the precise definition of a polymer's utility in biology. Think of it like a train. The entire train is the polymer, a large molecule made of repeating structural units.
The monomers.
The monomers. And in the case of nucleic acids, the monomers are nucleotides. So each boxcar on the train is a nucleotide.
Oh, I like that.
The elegance of this system is that the physical connection between the boxcars, the backbone of the polymer, can be identical and chemically completely stable.
But the contents of each boxcar can be different.
Precisely. The specific sequence of the individual nucleotide units can vary. The information isn't in the material itself, it's in the sequence of the units.
Going back to the computer analogy, it's the biological equivalent of binary code. The physical structure of the hard drive doesn't change shape. The hard drive is constant, but the arrangement of the ones and zeros holds all the data.
That is exactly how you need to visualize this. So the text introduces the two specific types of these nucleic acid trains, these polymers found in living systems. We have DNA and we have RNA, ribonucleic acid.
Right. Both polymers of nucleotides.
Both polymers.
Yeah.
But the text draws a very sharp distinction between their primary roles in the biological hierarchy. It states that DNA acts as the genetic material in most organisms.
In most organisms?
Yes. And listener, if you are preparing for exams, alarm bells should be ringing in your head right now. That distinction contains one of the most frequently tested concepts in competitive biology exams.
It's a classic trap.
The ultimate Neet trap. When a rigorous scientific text says most organisms, it is laying a trap by implying an exception. Students always want to say DNA is universal.
Because it's what we talk about 99% of the time.
But the immediate follow-up in the text is that RNA also acts as a genetic material, specifically in some viruses.
Which is just fascinating from an evolutionary perspective. For the vast majority of life, from a tiny bacterium all the way up to a blue whale, DNA is the ultimate stable archive. It's the master hard drive.
Right.
But viruses operate on the absolute edge of what we even define as biology. They strip away every unnecessary component just to maximize efficiency and replication speed.
They are the ultimate minimalists.
Yeah. So for some viruses, relying on DNA is evidently too cumbersome. Or perhaps using RNA provides a different evolutionary advantage for them.
And that forces you to be precise with your language. Imagine an exam question that states, DNA is the universal genetic material for all biological entities.
Sounds true at first glance.
But that statement is demonstrably false based on this exact text. RNA holds the primary genetic code for specific viral lineages. Understanding this exception is totally non-negotiable.
It proves nature isn't just monolithic. It uses whatever chemical pathway gets the job done.
Exactly. Now, the text then pivots to what RNA is doing in all the other organisms, the ones where DNA is the primary genetic material.
Right. So if DNA is the master archive holding the ones in use, the text argues that RNA is doing pretty much almost everything else.
This is your big realization, right?
Yeah, it really was a whoa moment for me. We always hype up DNA. But DNA just sits there. The text notes that RNA mostly functions as a messenger, but then it lists 3 other distinct roles. It says RNA acts as an adapter, structural, and catalytic molecule.
This multifunctionality of RNA is arguably the most profound realization in modern molecular biology, because we tend to view molecules through a very singular lens.
Right, like a molecule is either for structure, like cellulose in a plant wall, or it's for information like DNA.
Or it's for chemical reactions, like an enzyme. But the source text shatters that categorization completely. It presents RNA as a molecule that transcends all those boundaries. It wears multiple hats. Let's actually analyze the structural logic of these 4 hats, starting with its primary function as a messenger.
Okay, so the necessity of a messenger implies there's a separation between the storage of the information and the actual execution of that information.
Yes.
If DNA holds the master blueprint and the cell needs to physically build a protein based on that blueprint, it doesn't use the original DNA molecule directly. That would be risky.
Too risky. The DNA remains sequestered and protected.
Instead, the sequence is copied into a temporary mobile format, the messenger RNA. It's like taking a Post-it note copy of a specific page from an ancient, priceless spellbook. You don't take the book into the messy laboratory. You take the Post-it note.
The biological imperative here is preservation. The DNA archive is far too valuable to be subjected to the chaotic chemical environment of the cellular factory floor. By utilizing RNA as a messenger, the cell can send copies of the instructions out to be processed while keeping the original perfectly safe.
And it also allows for scale right. Amplification.
Crucial point. One sequence of DNA can generate thousands of temporary RNA messengers. That leads to rapid, massive scale production of whatever protein the cell needs at that moment.
Okay, so that's the messenger hat. The second role the text assigns to RNA is that of an adapter. Now, an adapter physically bridges 2 incompatible systems, like a travel plug adapter for your electronics.
Perfect analogy.
In the context of this cell, we have the information stored in the language of nucleic acids, the nucleotide sequence. But the final physical product, the protein, is built using an entirely different chemical language, the sequence of amino acids.
And this is a profound translation problem for nature. How does a sequence of nucleotides physically cause a specific amino acid to be placed in a chain?
Because they don't naturally stick together.
Exactly. There is no direct chemical affinity between a given nucleotide sequence and its corresponding amino acid. They don't interact. So the adapter RNA serves as the physical cipher. It bridges the gap.
One end of the adapter RNA plugs into the messenger RNA.
Yes, it recognizes a specific sequence.
But the other end is holding the correct amino acid. So it physically forces these two completely distinct chemical languages to interact in a predictable way.
It is the ultimate biological translator. Now, the text then mentions RNA's structural role, which is interesting because we usually think of information-carrying molecules as just linear strings, like a piece of ticker tape.
Just a sequence of data.
But RNA is a physical object in three-dimensional space. The sequence of nucleotides can actually cause the RNA strand to fold back on itself.
Because it's usually single-stranded, right?
Right. Unlike the rigidly paired double helix of DNA, which is basically locked into that one shape, RNA has the flexibility to adopt highly intricate globular shapes.
So these folded RNA structures can serve as actual physical scaffolding.
Yes, they form the physical architecture of large cellular machines. So the same type of molecule that carries the information is also physically building the factory itself.
That's amazing. But the fourth role, this is the one that completely upends classical biology for me. The text states that RNA functions as a catalytic molecule.
Ah, yes, the catalytic hat.
A catalyst actively drives chemical reactions. It lowers the activation energy. We're always taught from day one that proteins, specifically enzymes, are the cell's exclusive catalysts. Proteins do the work.
That is what makes a catalytic nucleic acid a conceptual earthquake. It means RNA is not just an inert piece of tape, and it's not just a structural beam. It is an active machine capable of breaking and forming chemical bonds.
Which is mind-blowing.
It really is. And for a Neet student listener, you must understand the gravity of this. Enzymes are not always proteins. RNA can act as an enzyme. And this single revelation actually solves one of the greatest paradoxes in all of evolutionary biology.
You mean the chicken and egg problem of the very first living systems?
Exactly. Unpack that for the listener.
Okay, so think about it. If you need DNA to store the instructions for making proteins, but you absolutely need proteins to catalyze the replication of DNA.
Neither could have existed first.
Right. Neither could exist without the other already being there. It's an impossible loop.
Which is why the text dedicates a specific section to the RNA world.
Yeah.
It's section 6.3 in the syllabus outline.
Oh, that makes so much sense now.
Right. If RNA possesses the ability to store sequence information, just like DNA does, and it can fold into physical structures, and it can catalyze chemical reactions like a protein,
then it's the ultimate multi-tool. It's the perfect candidate for the first self-sustaining biological molecule.
Precisely. The RNA World Hypothesis suggests a primordial era where RNA was the sole heavy lifter. It acted as both the genetic archive and the metabolic machinery.
Doing everything itself.
Yes. And then only eventually delegating the storage role to DNA, because DNA is more chemically stable. And delegated a catalytic role to proteins because amino acids are more versatile.
Looking at the sequence of the chapter outline provided in the text, you can really see how it builds this logical progression. It's not just a random list of facts. It constructs a narrative of how biological information flows.
It really does.
It starts with 6.1, the DNA, and 6.2, in search of genetic material, establishing the physical substrate and the 100-year ghost hunt. Then it moves to 6.3, the RNA world, which establishes the evolutionary versatility we just talked about.
And once that structural foundation is set, notice what the outline does. It shifts entirely to the active processes of the cell.
Right, from what it is to what it does.
The transition from section 6.3 to 6.4 marks the shift from structure to mechanism. It is mapping what biologists call the central dogma.
The central dogma of molecular biology.
Yes. The central dogma is simply the directional flow of genetic information. And to fully grasp this text, listener, you must understand why the outline is ordered this way. It is a strict chronological factory line.
Section 6.4 is replication.
Step 1.
Which is the absolute prerequisite for life to continue beyond a single cell. If the genetic material is the master instruction set, a cell cannot divide into two functional daughter cells without first perfectly duplicating that instruction set. You have to photocopy the blueprint before you build a second factory.
And the physics of replication requires an incredibly high degree of fidelity. The polymer has to be copied with near-perfect accuracy, because if the sequence of nucleotides is the information, then a change in the sequence is a corruption of the data.
A glitch in the code.
Exactly. While occasional mutations drive evolution over millions of years, on a cellular level, An error in replication can be lethal. The entire molecular basis of inheritance hinges entirely on this ability to create an exact physical replica of the DNA polymer.
Okay, so once replication ensures that the archive is secure and ready to be passed on, the cell needs to actually use the information to live its day-to-day life.
Right.
Which leads directly to section 6.5. The text explicitly defines this as the process of making RNA from DNA, which is transcription.
Consider the actual etymology of the word transcription. To transcribe is to copy information from one medium to another while remaining within the exact same language.
Like a court reporter. They transcribe spoken English into written English. It's still English.
Perfect. In the cell, transcription is the process of reading the sequence of the DNA polymer and synthesizing a complementary sequence of an RNA polymer. The language remains the exact same, the language of nucleic acids. You are simply moving the data from the highly stable, double-stranded archive of DNA to the temporary mobile format of messenger RNA.
This perfectly reinforces the concept of RNA as the messenger hat that we discussed earlier. The transcription process isolates the valuable DNA from the wear and tear. The RNA transcript is sent out to the factory floor, and if it gets chewed up or degrades after use, it doesn't matter.
Because the DNA master copy is sitting safely in the nucleus, ready to generate more transcripts whenever needed.
Exactly. But the output of transcription is just a sequence of RNA nucleotides, and the cell actually requires proteins, which are polymers of amino acids.
So we have a language barrier.
A massive language barrier, which requires a shift from transcription to translation. But before you can translate, you must understand the rules of the translation.
Which is why section 6.6, the genetic code, sits precisely between transcription and translation in the outline. It has to be there logically.
The genetic code is the cipher. The text says the genetic code determines the sequences of amino acids in proteins. It's the mathematical solution to the translation problem we talked about.
Right.
You have a sequence written in an alphabet of only 4 nucleotide bases, and you somehow need to translate that into a sequence using an alphabet of 20 completely different amino acids.
The existence of the genetic code is proof of a universal biological logic. It is a highly specific, rigid set of rules detailing exactly how a sequence of nucleotides dictates the selection of amino acids.
Without the genetic code, the RNA messenger is just a meaningless string of chemicals.
Exactly. The code is what gives the physical molecule its informational value. It is the dictionary the cell uses to read the nucleic acid script and understand exactly what protein needs to be built.
So armed with the messenger RNA from transcription and the dictionary of the genetic code, the cell can finally execute section 6.7, translation, which the text explicitly defines as the process of protein synthesis.
And again, look at the word. The term translation is chemically accurate here.
Yeah.
You are changing languages completely. You are reading the sequence of the RNA polymer and using that information to synthesize an entirely new type of polymer, a polypeptide chain, which will eventually fold into a functional protein.
And this is where those adapter RNAs come into play.
Yes, the adapters are physically bringing the correct amino acids to the assembly line, plugging into the messenger RNA based on the sequence. Translation is the culmination of the entire genetic process.
It's the physical payoff.
It is the exact moment the abstract information stored in the DNA becomes the physical working machinery of the organism.
If we map this out chronologically, replication copies the DNA.
Check.
Transcription makes an RNA copy from the DNA.
Check.
The genetic code provides the translation dictionary. And translation uses the RNA to synthesize proteins. It is a highly ordered sequential manufacturing pipeline. But the outline doesn't stop at translation, which is interesting. The next topic, section 6.8, is regulation of gene expression.
The necessity of regulation becomes totally obvious when you consider the economy of a living cell. Think about a multicellular organism like yourself, listener. Every single cell in your body contains the exact same DNA master archive.
Right. A neuron in my brain contains the exact same genetic instructions as a muscle cell in my heart.
Exactly. Now, if the transcription and translation pipeline just ran constantly and uniformly in every cell would produce every possible protein simultaneously.
Oh, wow. So the heart cell would be trying to build brain receptors.
It would be absolute biological chaos, a massive waste of energy and resources. The cell would destroy itself.
So the regulation of gene expression is the control system. It's the volume knob. It determines which sections of the DNA are actually transcribed into RNA and when and in what quantities.
It is the mechanism that allows a cell with a universal instruction manual to specialize. It turns specific genes on when their corresponding proteins are needed and completely silences them when they are not.
Without precise regulation, the sheer complexity of multicellular life is impossible. Regulation is how an organism responds to its environment, how it manages its metabolism, and how it guides its own development from a single fertilized egg into a complex adult.
The flow of information isn't just about the capability to make proteins. It is about the sophisticated control of that capability.
Okay, so the outline up to section 6.8 provides a complete theoretical framework for how life operates at the molecular level. It covers the physical structures, the functional processes, and the control mechanisms.
The complete central dogma.
But then the text undergoes a massive shift in scope and timeline. The final two sections of the chapter outline move from fundamental cellular biology to modern applied science.
The transition from section 6.8 to 6.9 marks the passage from understanding the mechanisms of the code to actually reading the entirety of the code itself. Section 6.9 is the Human Genome Project.
And the text frames this as a monumental milestone. It states, the determination of complete nucleotide sequence of human genome during last decade has set in a new era of genomics.
The phrase complete nucleotide sequence is staggering when you really consider the scale of what that means. We established earlier that the information is held in the sequence of the polymer, the sequence of the boxcars on the train.
Right.
The human genome consists of billions of these nucleotide monomers linked together. Billions.
Which puts that 100-year gap into perspective again. For the entire century following Mendel, the goal was just to identify what the genetic material even was and how it functioned, basically. The Human Genome Project represents the moment humanity decided to actually sit down and read every single letter of the Human Instruction Manual.
It is a profound philosophical turning point in science. Prior to this, biology was largely observational. It focused on isolated genes and their specific localized effects. Genomics, the new era heralded by this text, is the study of the entire integrated genetic system as a whole.
Because once you determine the complete nucleotide sequence, you are no longer guessing at the abstract factors Mendel described.
No guessing involved. You possess the literal chemical text of the software that runs human biology.
And possessing that complete sequence naturally leads directly to technological applications. Which brings us to the final section of the outline, 6.0, DNA fingerprinting. The text specifically notes that the chapter will discuss the consequences of human genome sequencing, and fingerprinting is the most direct consequence of understanding sequence variation.
DNA fingerprinting is the ultimate practical validation of the molecular basis of inheritance. If every individual's DNA is a massive polymer, and the sequence of that polymer determines the individual's unique traits,
then subtle variations in that sequence must exist between different individuals.
Exactly. By analyzing specific, highly variable regions of the complete nucleotide sequence, scientists can generate a molecular profile that is utterly unique to an individual.
It takes the profound, abstract concepts of replication, transcription, and translation and turns them into a practical tool. A tool that can definitively identify a specific human being from a microscopic biological sample left at a scene.
It closes the loop entirely. We started with Mendel, who couldn't even see the factors, and we end with a technology that reads the physical molecules so precisely that it serves as an infallible biological barcode.
The structure of this chapter's outline is really a masterclass in scientific narrative when you lay it all out like this. It forces the learner to walk the exact historical and logical path of discovery.
Yes. You begin with the chemical definition of the material. You explore its functional capabilities through the versatility of RNA. You map the strict chronological flow of its information. You understand the necessity of its regulation. And finally, you grapple with the implications of humanity possessing the complete sequence.
For someone studying this material, particularly someone preparing for high-stakes evaluations like Neet, the challenge isn't just memorizing these headings. It's understanding the logical necessity connecting them. If you understand why transcription physically must precede translation, the facts just organize themselves in your mind.
Absolutely. And to solidify that logical structure for you, the listener, we need to explicitly anchor the core concepts we have dissected today. If you are analyzing this text, there are absolute non-negotiables you must retain. Let's do the memory anchors. Here is what you must remember regarding the fundamental chemistry.
The foundational structure of the genetic material.
Precisely. You must remember that nucleic acids are polymers of nucleotides. The polymer is the long, continuous chain, and the nucleotides are the individual, repeating chemical units that form it. The sequence of these nucleotide units is the physical embodiment of genetic information. That's anchor number one.
Anchor number two is regarding the division of labor between the nucleic acids, and the text is very specific about exceptions here.
Yes. Here's what you must remember regarding DNA and RNA. DNA acts as the genetic material in the vast majority of organisms, providing a highly stable archive. However, the critical exception to always monitor for exams is that RNA acts as the primary genetic material in some viruses. Never assume DNA is universal without acknowledging that viral exception.
Anchor number 3. Beyond storing genetic code in viruses, the text completely redefines the role of RNA in the rest of biology.
Here is what you must remember regarding the versatility of RNA. It is not merely a passive carrier of information. RNA performs 4 distinct critical functions. It acts as a messenger, ferrying the transcribed code. It acts as an adapter, physically bridging nucleotides and amino acids during translation. It serves a structural role, folding into the physical scaffolding of cellular machinery. And most profoundly, it can act as a catalytic molecule, actively driving chemical reactions, which provides the foundation for the RNA World Hypothesis.
And anchor number 4 is the roadmap of information flow, the central sequence of the chapter outline.
Here's what you must remember regarding the flow of genetic information. The central dogma. The sequence is rigid. First, replication perfectly duplicates the DNA. Second, transcription reads the DNA to synthesize messenger RNA. Third, the genetic code provides the mathematical translation rules. Fourth, translation utilizes that code to synthesize the final physical protein. And fifth, regulation of gene expression controls this entire pipeline.
Finally, anchor number 5. The text shifts to the modern implications of this knowledge.
Here is what you must remember regarding the era of genomics. The Human Genome Project successfully determined the complete nucleotide sequence of the human genome. This transition enabled powerful applications, most notably DNA fingerprinting, which relies on analyzing the unique variations within that complete sequence.
Okay, let me try to give a rapid-fire sticky summary of everything we've covered. 60 seconds, here we go.
Go for it.
Over 100 years, we went from Mendel's mysterious invisible factors to discovering that DNA and RNA are physical polymers of nucleotides. We learned DNA holds the master code, but RNA acts as the messenger, adapter, structural scaffolding, and even a catalyst. This genetic code flows chronologically from replication to transcription, utilizing the genetic code dictionary, to translation to actually build the proteins that make you who you are, all carefully regulated by the cell. And now, thanks to the Human Genome Project, we can actually read that whole sequence from start to finish, which gives us tools like DNA fingerprinting.
Perfect summary. Synthesizing all of this really provides a complete perspective on the molecular basis of inheritance. The transition from Mendel's abstract math to reading the complete human nucleotide sequence represents arguably the greatest technical achievement in the history of biology. The text provides the exact roadmap of how that physical system operates.
It really does.
But listener, it leaves us with a deeper scientific reality to confront, something to ponder as you close your books today. We spent a century trying to find the hard drive. We spent the last decade reading every single line of code written on it. We now understand exactly how the RNA transcribes it and how the cellular machinery translates it into the physical structure of a human being. The science of genomics is no longer about discovering what the code says. The frontier we are currently crossing, the question you will have to answer in your generation of science, is determining what happens when we possess the tools to actually edit the sequence ourselves.
That's a heavy thought to end on. Thank you, listener, for joining us on this deep dive. Keep asking those foundational questions.
Keep exploring. We'll see you on the next deep dive.